All right, today we're going to unpack a really brilliant piece of biohacking. It's a story about how scientists took a cue from a cell's emergency survival kit to solve one of modern medicine's biggest headaches. Super clever stuff. It really all starts with a question that's been bugging researchers for decades. We have these incredible life-saving medicines made from proteins. Things like insulin or those super complex antibodies. But actually making them efficiently can be a total nightmare. So why is that?
Okay, so for about 50 years now, the basic idea has been pretty simple, right? We take the genetic blueprint for a human protein, let's say insulin, for example, and we pop it into a fast-growing bacterium like E. coli. And just like that, these little microbes become tiny living factories, just churning out the protein we need. This is literally a cornerstone of the whole biotech industry.
But of course, there's a huge catch. When you try to crank up the speed on these little factories, well, that's when things can go very, very wrong. You see, a protein is only useful if it's folded into a very specific, perfect 3D shape. I love this analogy. Think of it like a perfectly folded origami crane. That's a functional protein. But when the cellular factory is rushed, it doesn't take the time to fold it correctly. Instead, it just crumples it up into a useless ball of paper. And in biology, these crumpled up, totally non-functional clumps have a name: inclusion bodies. And this is the core of the problem, right here. We push the bacteria to make protein as fast as humanly possible, but that speed just leads to misfolding. All these proteins clump together into those useless inclusion bodies. Now, you can try to unfold and then refold them, but that process is incredibly expensive and super inefficient. So a huge amount of what we're making just ends up as garbage. So, okay, how do we fix this mess? Well, the answer, funnily enough, came from a totally different question. Instead of asking how to make a cell go faster, some clever scientists started asking, what happens when you slam on the brakes? Okay, just imagine for a second. You're a bacterium. You're just happily swimming around at a cozy 37 degrees Celsius. That's human body temp. Then, all of a sudden, bam, the temperature plummets to a freezing 15 degrees. This is a life or death emergency. So what on earth does the cell do to survive? It triggers this really remarkable survival protocol called the cold shock response. The first thing it does, it slams on the emergency brakes. All non-essential protein production just stops. Then, in that suddenly quiet cellular environment, it rapidly pumps out a very specific set of proteins called, you guessed it, cold shock proteins, or CSPs for short. And once those are ready, the cell can finally adapt and slowly start growing again in the cold. So the really crucial point is this. The cell has a special genetic on switch. It's called a cold shock promoter, and it flips on only when the temperature drops. At normal temps, it's off, but when it gets cold, it flicks on. It's a beautifully precise, temperature-controlled switch. So what's the job of these special proteins anyway? Well, they act as something called RNA chaperones. The best way to think of them is like cellular project managers. Their job is to organize all the cell's machinery and make sure everything is still working efficiently, even in that stressful, cold environment. They basically bring order to the chaos. And this is where our two stories collide. On one hand, you have the protein production problem. It's fast, it's chaotic, it's messy. And on the other, you have nature's solution to cold stress: slow, orderly, and precise. The aha moment was when scientists realized they could use one to fix the other. Honestly, the big idea was just breathtakingly clever. It was, what if we could hijack that genetic cold shock switch? Instead of having it turn on a cold shock protein, what if we could rig it to turn on the protein medicine we actually want to produce? So here's how the hack works. On the left, you've got nature's original setup. The cold shock promoter is wired to the gene for cold shock protein. Simple. But on the right, check out the engineered version. Scientists use genetic tools to literally snip out that original gene and swap in the gene for a human medicine. And just like that, that temperature-sensitive switch is now in control of our factory. Okay, so the theory is beautiful, but the big question is, does it actually work in practice? Well, the results are pretty stunning. The answer is a resounding yes. For example, a 2020 study was looking at this one protein called PACRG. They found that the cold shock system, which is commercially known as P-cold, was dramatically more effective than a standard method, the PET system. And this quote really says it all: 15 to 20 times more protein. Not 15% more,
15 times more. And this chart really drives the point home. That tiny little bar on the left, that's the amount of soluble, correctly folded protein you get from a standard system. Now compare that to the massive bar on the right. That's the output from the cold shock system. The difference is it's staggering. I mean, really, let that number sink in for a second. A 15 to 20-fold increase in the useful stuff, the good product. This isn't just some tiny incremental improvement. This is a complete game changer, especially for making those proteins that were always considered difficult to express.
So, to sum it all up, the advantages here are huge. First, you get way more active, usable protein. Second, you completely avoid making those useless inclusion bodies. It even works for proteins that are normally toxic to the cell at warmer temperatures. And because all you have to do is lower the temperature instead of adding a bunch of expensive chemicals, it's a much cleaner, greener process. The industry actually has a cool name for this: white biotechnology, using living cells to make things in a more sustainable way.
So look, this is already a massive success in bacteria like E. coli. But the story definitely doesn't end there. The next frontier is, I think, even more exciting.
You see, bacteria are fantastic for making simple proteins, but today's most advanced medicines, we're talking complex antibodies and things like that, they're far more complicated. They need the super sophisticated cellular machinery you only find in hosts like yeast or even mammalian cells to be folded correctly. So the cold shock principle is already proven in bacteria, but now it's starting to emerge as a powerful tool in these more advanced systems too. And this could unlock a whole new wave of complex therapies that right now are just too difficult or expensive to produce at scale.
It really makes you wonder, doesn't it? The solution to a multi-billion-dollar manufacturing problem was just hiding in a bacterium's response to getting a little cold. It's just a perfect reminder that nature is the ultimate engineer. And it leaves you with this one big question: what other secrets are waiting in its survival kit for us to discover next?